Hello and welcome. Today let's continue with the second chapter. We'll move on to the biochemistry aspect of this chapter now. And here this part two, will take us into, like I said, biochemistry. Now when we talk biochemistry, we can also refer to biochemistry as biological chemistry. Biological chemistry. Now, when we talk biological chemistry or biochemistry, I'd like you to know that biochemistry is the study of the chemical compositions, or you can say the chemical components. So it is the study of the chemical compositions and reactions of living matter. Now, that's one way we could describe biochemistry or define biochemistry. Another way we can define biochemistry is as the study of chemical processes. We can also define biochemistry as the study of chemical processes within and relating to living organisms. So the study of chemical processes within and relating to living organisms. Now I'd like you to know all chemicals in the body are either organic compounds. All chemicals in the body are either, number one, organic compounds, or number two, inorganic compounds. Now, when we talk organic compounds, so all chemicals in the body are either organic or inorganic. Now, when we talk organic compounds, I'd like you to know the organic compounds, they contain carbon. And they are all covalently bonded molecules. And many we're going to see are large molecules. Now, when we talk inorganic compounds, our inorganic compounds will be all other chemicals in the body. This will include water, salts, and many acids and bases. Now, both organic and inorganic compounds are equally essential for life. They are both equally essential for life. Let's talk first. So when we said inorganic compounds, water, salts, and many acids and bases, and they do not contain carbon in their backbone. While organic compounds, they do contain carbon, we said. And when we talk organic compounds will include carbohydrates, fats, proteins, and nucleic acids. And again, we said they will contain carbon in their backbone. They're usually large molecules, and they are also usually covalently bonded molecules. Again, both equally essential for life. Next, then, let's move down and let's talk more about our inorganic compounds. Now, when we talk inorganic compounds, we'll focus on our acids and bases. Now, when we talk about our acids and bases, I would like you to know, when we talk about our acids and bases, I'd like you to know both can ionize and dissociate in water. Acids and bases both can ionize and dissociate in water. Acids and bases can conduct electrical currents, but we won't really go into studying that. Let's talk first acids. Acids, I would like you to know, have a sour taste, and they can react with many metals, and they can even burn a hole in your rug. They're that potent. An acid is a substance that releases hydrogen ions. It releases hydrogen ions. So when I'm saying hydrogen ions, a hydrogen ion, and here you can see it's indicated with a plus. So if you recall, that's just a bare proton. That means it's given up its negative charge, its electron. So with the electron being gone, it's just a proton that's going to remain. So that's why we refer to it as a bare proton, or we can refer to it as a hydrogen ion. So an acid is a substance that releases hydrogen ions in detectable amounts when dissolved in water. So a hydrogen ion is just a hydrogen nucleus or a naked proton you can think of, right? Acids are also defined as proton donors. Acids are also defined as proton donors. When acids dissociate or dissolve in water, they release hydrogen ions or protons and anions. So when acids dissociate or dissolve in water, they release hydrogen ions or protons and anions. The concentration of protons then determines the acidity of a solution. So we measure the quantity of protons, and that determines the acidity of a solution. Anions, the anions have no effect on acidity. Here we can see our first example. Our first example is hydrochloric acid. And when we talk hydrochloric acid, when it dissociates, you can see HCl. When hydrochloric acid dissociates, it dissociates into a proton, we see here, and a chloride ion, and into a chloride ion. So here you have your proton, and the chloride ion is our anion. That's the anion. We said this has no effect on the acidity. This here will have an effect on the acidity. The proton will, the hydrogen ion will. Important acids, hydrochloric acid we just saw there. Carbonic acid, H2CO3. Carbonic acid used in soft drinks, so H2CO3, when it dissociates now, it's going to dissociate, and when it dissociates here, we'll see, let's write this on here. I can get the pen. So we have H2CO3. And now when it dissociates, so we're going to see a hydrogen ion, a proton being released, and along with that we're going to have a bicarbonate ion. HCO3, right? We're going to have a bicarbonate anion released there as well. So here's the proton that's released, and then the bicarbonate ion, just like we saw here, the anion. Here's our anion. We also have, we can see here, acetic acid. Acetic acid is in vinegar, right? Acetic acid, when it releases a hydrogen ion, a proton, right, it's going to release then one proton, and acetate ion, and an acetate ion. So here we'll have HC2H3O2. So here what gets released? One hydrogen ion, and we've got C2H3O2. H2. Okay, so here acetate. Okay, so here we have acetic acid. Acetic acid, when it dissociates into a proton and also the anion, which is our acetate ion, which is the anion. Next, then here we're going to see we've got our bases. Now, when we talk bases, bases are proton acceptors. They are proton acceptors. They take up hydrogen ions. They take up hydrogen ions. They're going to take up hydrogen ions from the solution. Now, common bases will include hydroxides, such as magnesium hydroxide, milk of magnesia, right? MgOH. Okay, we don't have that listed here. Here we have sodium hydroxide. MgOH then would be milk of magnesia. Here we have sodium hydroxide. This is lye for washing, cleaning, and in Drano. Hydroxides also dissociate when dissolved in water. But here, hydroxyl ions, the OH, and cations, the NH we see here, are liberated. For example, of ionization of sodium hydroxide we see here is going to yield sodium as our cation, and then here we have our anion, our hydroxyl ion. So OH now is able to accept an available proton. And you all know what then H plus OH is going to give us. We'll have H2O, water. So when we talk bases I described as proton acceptors. They take up hydrogen ions. And that's exactly what we see here. They take up hydrogen ions. So now this reaction produced water, which helped to reduce the acidity of the solution. Bicarbonate ions we see here HCO3. Bicarbonate ion is going to be an important base in the body. These bicarbonate ions are going to be very important bases in the body and are very abundant in the blood. Ammonia, NH3 we see here, is a waste product. It's a waste product of protein breakdown. It's also a base. And it attracts protons. When it attracts protons, then you'll see NH3 then, let's erase all of this here. When it attracts a proton, NH3 is going to become NH4. It will become NH4, right? Another hydrogen gets added because the hydrogen that was in the solution causing the solution to be acidic gets picked up. So bases help to decrease the acidity. So they're proton acceptors. Okay. Ammonia we saw here is a base and it's going to attract protons, helping to form an ammonium ion. Next time we talk about pH. Potential hydrogen, pH. pH deals with acid-base concentration. It deals with acid-base concentration. Now, when we talk pH, we'll see the more hydrogen ions that we have in a solution, the more acidic the solution is. The more acidic then the solution is. The greater concentration of hydroxyl ions, the more basic or the more alkaline we say that the solution becomes. Now, the concentration of hydrogen ions is measured as pH units, potential hydrogen units. So the pH scale checks the concentration of hydrogen ions in a solution. The pH scale checks the concentration of hydrogen ions in a solution. The pH scale, I want you to know, runs from 0 to 14. It runs from 0 to 14. 1, 4, to 14. At pH of 7, the solution is considered neutral. At a pH of 7, the solution is considered neutral, meaning the solution is neither acidic nor is the solution basic. The number of hydrogen ions equals the number of hydroxyl ions in a neutral solution. So in a neutral solution, a solution that is neither acidic or basic, the number of hydrogen ions equals the number of hydroxyl ions. Distilled water is an example. Distilled water is an example of a neutral solution. Distilled water has a pH of 7. Solutions with a pH below 7 are considered acidic solutions. In acidic solutions, the hydrogen ion quantity will outnumber the hydroxyl ion quantity. The lower the pH, the more acidic the solution is. A solution with a pH of 6 has 10 times as many hydrogen ions as a solution with a pH of 7. Solutions with a pH higher than 7 are alkaline, and the hydrogen ion concentration decreases by 10 with each pH unit. For example, solutions with a pH value of 8 and 12. Okay, so let's say we have two solutions, one with a pH value of 8 and one with a 12. Solutions with a pH value of 8 and 12 have a 1/10 and a 1/100,000th as many hydrogen ions as a solution with a pH of 7. So some examples: neutral pH, we said, is going to be distilled water. And here you can see solutions that have an acidic pH, from milk to coffee, black coffee to wine, down to lemon juice and gastric juice, we're going to talk about later on in the semester, down to hydrochloric acid, which helps to make up the gastric juice. And then we talk basic solutions. You can see here. Here we've got blood 7.4. Blood ranges from 7.34 to 7.45. So basic solution there. Then egg whites, household bleach, 9.5 pH. And then we've got ammonia, oven cleaner, lye, all the way to sodium hydroxide. So examples of basic solutions. And then down here we've got examples of acidic solutions. And then at a pH of 7, we have neutral solutions. All right, let's move next and let's move to buffers. Let's talk buffers. Now, when we talk buffers, I'd like you to know buffers are substances. They are substances that prevent, or you can say resist, abrupt and large swings in the pH of body fluids. This is done by releasing hydrogen ions, acids, right? When the pH begins to rise, and by binding hydrogen ions, acting as a base, when the pH drops. So again, buffers are substances that prevent or resist abrupt and large swings in the pH of body fluids. And again, this is done by releasing hydrogen ions, okay, when the pH begins to rise, and by binding hydrogen ions when the pH drops. Now, normal blood pH varies from 7.35, we said, to 7.45. It's very a minute change, 0.1 of a change, and even that is sometimes unbearable for the body. So our body has one major buffer system that I'd like you to know about that helps to maintain pH homeostasis of blood. That buffer system is the carbonic acid bicarbonate system. Here, carbonic acid, we see down here H2CO3, it dissociates reversibly. When it dissociates reversibly, it releases bicarbonate ions and protons. So here you can see, if the blood becomes too basic, you have a right shift. If it becomes too basic, you have a right shift. If blood becomes too basic, you have a right shift. You have a right shift, and what that does, it helps to release then protons. It releases protons, which then acidify our blood. Now, if our blood is too acidic, then we're going to have a left shift. We'll have a left shift, and then what happens there is these bicarbonate ions will start to pick up these hydrogen ions, making our blood then more basic. So if the blood becomes too acidic, then we have a left shift. And by picking up hydrogen ions, this is going to make our blood more basic. This chemical equilibrium between carbonic acid and bicarbonate ions, it helps us to resist changes in blood pH by shifting to the right and to the left as hydrogen ions are added or removed from the blood. Here we can appreciate then some common functional groups that I'd like you to know about and be familiar with. Here we've got carboxyl group, COH, hydroxyl group that we've gone through, we've talked about, and a phosphate group. Next, then let's move on to our organic compounds. Now, when we talk about our organic compounds are going to be molecules that are unique to living systems, and they will include carbohydrates, as we have mentioned, lipids, our fats, proteins, and nucleic acids. Each, we said, contains carbon as a backbone in its structure, while inorganic compounds are defined as compounds that lack carbon, we said. But there are a few exceptions that do exist, but we won't get into it that much detail. Now we'll see many biological molecules, carbohydrates, and proteins are polymers. Polymers are chain-like molecules. They are made up of many similar or repeating units, or you could say made up of many monomers, which are joined together, we're going to see via dehydration synthesis reactions. So when these monomers are joined together via these dehydration synthesis reactions, this helps to form polymers. So we'll go through and we'll check out all these different polymers and monomers that build them up as well. So dehydration synthesis, and then the reactions that are going to help break them down will be our hydrolysis reactions. So let's see here when we talk dehydration synthesis reactions. During dehydration synthesis, a hydrogen atom, okay, you can see here, a hydrogen atom is going to be removed from one monomer, and a hydroxyl is removed from another, giving rise to then water, and also helping to unite the two monomers, forming then a dimer, we can say, right? Two monomers linked together. Now, opposite, we're going to see this happen in the digestive tract. There, these monomers that have been put together now that have become a dimer, those dimers now don't get absorbed as dimers. They have to be broken down to the monomer. So there what happens is water gets added to those dimers, and when they break apart, you have then the formation of your two monomers, the constituent building blocks. That's what then gets absorbed. So here we could see a dehydration reaction putting glucose and fructose together, forming sucrose, we're going to talk about in a few minutes. And then we break down, because we can't absorb sucrose, so sucrose has to get broken down to its monomers, okay, which are the monosaccharides here, and that's a disaccharide, sucrose is. So the monosaccharides then are going to be formed via the hydrolysis reaction, where we add water to our dimers, forming then our building blocks or our constituent building monomers. Let's talk carbohydrates. Now, when we talk carbohydrates, carbohydrates are going to be a group of molecules that includes sugars and starches. Carbs can contain carbon, hydrogen, and oxygen in their structure. They can be classified according to the size. Carbohydrates can contain carbon, hydrogen, and oxygen in their structure. They can be classified according to their size and solubility as monosaccharides, which are simple sugars or one sugar, disaccharides, two sugars, or as polysaccharides, where we're talking about many sugars. So let's talk first about monosaccharides. Now, when we talk monosaccharides are also known as simple sugars. They are also known as simple sugars. Monosaccharides are the building blocks of other carbohydrates. And they are a single chain or single ring structures. And they are single chain or single ring structures that contain anywhere from 3 to 7 carbon atoms, as we can see here in this picture. Monosaccharides are generally named according to the number of carbon atoms they contain. There are two important ones in the body, and they include our pentose or 5 carbon sugars and our hexose or 6 carbon sugars. Now, when we talk about our pentose sugars, examples of our pentose sugars will be ribose, we see here, and deoxyribose, we see here. I also want you to know the difference between the two, and the name is telling you everything, but I want you to know where that oxygen is lacking. It's the second carbon. When we talk about then our hexose sugars you can see include galactose, fructose, and glucose. So glucose, fructose, and galactose. Next then, let's move to our disaccharides. Now when we talk about our disaccharides are double sugars. Disaccharides are formed when 2 monosaccharides are going to be joined together via our dehydration synthesis reaction. And I told you they're too large to pass through cell membranes, so they have to be broken down in order to be absorbed. Now, when we go through and we talk about our disaccharides, examples will include sucrose, maltose, and lactose. So first we talk sucrose. Sucrose, you need to know, is formed when glucose joins together with fructose. They give rise to sucrose. We talk maltose. Maltose, then you can see malt sugar. Sucrose is table sugar, right? It's maltose, malt sugar, is formed when glucose comes together with another glucose. So 2 glucose units come together. Now when glucose and galactose come together, we form milk sugar, lactose. Milk sugar. Maltose, malt sugar, and candies, Whopper candies, or even liquor as well. Malt drinks. So again, they're too large to pass through cell membranes. So they're going to be digested to their simple sugar units to be absorbed in the blood from the digestive tract. Next, then we have our polysaccharide. When we talk about our polysaccharides are many simple sugars linked together. Because they are large and insoluble molecules, they are ideal storage products. Starch and glycogen are of major importance to the body. Both starch and glycogen are polymers of glucose. The amount of branching is the only difference between the two. Starch is a storage carbohydrate that's formed by plants. When we eat starchy foods, such as grain products or potatoes, right, pasta, rice, the starch has to be digested for its glucose to be absorbed. Cellulose, another plant polysaccharide, doesn't get digested by us. We don't have the enzymes to break it down. And what it does is it gives plant cell walls strength. It gives plant cell walls strength. That's cellulose. That's like corn, peas, right? These foods, they'll come out basically then looking like when they went in. So here we can see glycogen. Glycogen, the storage carbohydrate of animal tissues. It's stored in skeletal muscle and liver cells. Now, when blood glucose sugar levels drop sharply, liver cells have the ability to break down this glycogen through a process we call glycogenolysis and release its glucose into the blood. So now when we have this new glucose in the blood, We call that gluconeogenesis. That's the generation of new glucose. So glycogen can be broken down. So glycogen can be broken down to release glucose. When we talk about carbohydrate function, the major function is to provide a ready, easily used source of cellular fuel. And we'll see glucose is at the top of the menu for our cells. In our bodies, glucose is broken down and used to synthesize ATP. Now, when ATP supplies are sufficient, dietary carbohydrates are converted to glycogen, which we saw here, or to fat and stored then in the body. Carbohydrates are also found attached to the external surface of our cells. And they will also function as structural molecules. All right, next then let's talk about lipids. We'll talk about lipids. When we talk lipids, I'd like you to know lipids are going to be insoluble in water. And just like carbohydrates, they also are going to contain carbon, hydrogen, and oxygen in their backbone. Lipids will include triglycerides, our fats and oils, phospholipids, steroids, and a number of other substances. Let's talk first our triglycerides. Now, when we talk about our triglycerides or our neutral fats, our triglycerides, our neutral fats, are known as fats when solid or oils when liquid. They are composed of two types of building blocks, fatty acids and glycerol molecules. The fatty acids are linear chains of carbon and hydrogen atoms, basically hydrocarbon chains. In fat synthesis, three fatty acid chains are attached to a glycerol molecule by dehydration synthesis. The result then is going to be an E-shaped molecule we see here in this picture. So here's the glycerol molecule and the three fatty acids. And when they come together, thanks to the dehydration synthesis reaction, you can see here we form an E-shaped molecule, the letter E. The glycerol backbone is the same in all triglycerides. But the fatty acid chains vary, resulting then in different kinds of fats and oils. Triglycerides are large molecules, often consisting of hundreds of atoms. Ingested fats and oils must be broken down to their building blocks before they can be absorbed, just like we saw with our polymers in relation to carbohydrates. The hydrocarbon chains you see here, they're going to make the triglyceride molecules non-polar, because polar and non-polar molecules don't interact. Oils or fats and water don't mix, and water is polar. Triglycerides, they provide large amounts of energy. They also function to insulate and cushion and protect our organs. Fatty acids that have only single covalent bonds between the carbons. So these fatty acids that have only single covalent bonds between the carbons are called saturated fatty acid chains. So again, fatty acids that have only single covalent bonds between the carbons are called saturated. Their fatty acid chains, you can think of, are saturated with hydrogens. They have the maximum amount of hydrogens that they can contain. And there's no double bonds you'll see in those fatty acid chains. There are no double bonds in those fatty acid chains. Next, you have the saturated fatty acid chains. Now, saturated fatty acid chains are straight. Saturated fatty acid chains are straight. They have no kinks or bends in them at all. And at room temperature, the molecules of saturated fat, are packed together, forming a solid. Fatty acids that contain one or more double bonds between the carbon atoms. So if we were to have a double bond here and a double bond here or a double bond here, so again, fatty acids that contain one or more double bonds between the carbon atoms, they're said to be unsaturated. They are said to be unsaturated. Then we could complicate that even more, and we can have monounsaturated, where there's one double bond, or polyunsaturated, where there's many double bonds in between the carbons. Now, we talk monounsaturated. Okay, we're talking sunflower, safflower oil. We're talking polyunsaturated. Okay, we're talking omega-3 fatty acids, omega-6 fatty acids. These are all fatty acids that are heart healthy. This will include then, you'll see, now the difference between omega-3 fatty acids, omega-6 fatty acids is the distance. It's the distance between the beginning of the carbon chain and the first double bond. So that's why we have omega-3s and omega-6s. Now again, they're heart healthy, so this will include fatty fish, okay, canola oil, walnuts, sardines, mackerel, salmon. These fats, they are heart healthy, and they prevent and they treat heart disease and stroke. These are heart healthy fatty acids, and they prevent and treat heart disease and stroke. So here, when we have an actual double bond, this is what it'll look like. So you see double bond here, it causes that fatty acid chain not to be straight. It instead now has a kink, a bend in it. So the double bonds cause the fatty acid chains to kink or bend, so they cannot be closely packed enough to solidify. So you'll see triglycerides with short fatty acid chains or unsaturated fatty acid chains are oils. They're oils. They're a liquid at room temperature. And they are typically of plants. So saturated, unsaturated we saw there. Now, longer fatty acid chains and more saturated fatty acids are common in animal fats. Longer fatty acid chains and more saturated fatty acids are common in animal fats such as butter fat and the fat of meats, which are a solid at room temperature. Now, of the two types, saturated and unsaturated, of the two types of fatty acids, the unsaturated variety, especially olive oil, is said to be more heart healthy. Trans fats, then we have next. Trans fats or modified oils, they're common in many margarines and baked products. Trans fats are oils that have been solidified by the addition, again, trans fats are oils that have been solidified by the addition of hydrogen atoms. They have added hydrogen atoms at the sites of the double bonds. So it breaks the double bonds and adds hydrogens there. Trans fats, they're said to increase the risk of heart disease. So that's why you see now most food products will have signs on them saying no trans fat, trying to promote people to consume them. And then again, when we talk omega-3 fatty acids, more heart healthy. Let's talk then phospholipids. Phospholipids are modified triglycerides. They are a modified triglyceride. Specifically, they are a diglyceride with a phosphorus group. Specifically, they are a diglyceride with a phosphorus group and two, not three, but two fatty acid chains. The hydrocarbon tail is non-polar. The hydrocarbon tail is non-polar and hydrophobic, meaning it does not like water. The phosphorus containing head is polar and it is hydrophilic. It attracts charged particles like water or ions. Since it's got this polar and non-polar end, this means that this molecule is amphiphatic. It is amphiphatic, meaning they have both hydrophobic and hydrophilic sides. This characteristic allows them to be used as the chief building material for cell membranes. Third, then we have our steroids. So when we go through, we talk cell membranes, we're going to have actually a double layer of these phospholipids. So one, you can see phospholipid basically lying this way, and then another one is going to be lying this way. We're going to have a phospholipid bi-layer when we look at that in the next chapter. Next then, our steroids. Steroids are fat-soluble. This allows them to pass through the cell membrane. Steroids are fat-soluble. This allows them to pass through the cell membrane. They have a different structure from other lipids. When you look at their structure is basically four interconnected rings. Four interconnected rings of carbon atoms you see that form the skeleton of our steroids. The single most important molecule is cholesterol. It's cholesterol. Then after cholesterol, you'll have cortisol, estrogen, and testosterone. Steroids fall in the hormone category. We ingest cholesterol in animal products such as eggs, meat, and cheese. Also, our liver produces minute quantities of cholesterol as well. Even though cholesterol has a bad name because of its role in atherosclerosis, it is very essential to life. It is essential to life. Cholesterol is found also in the cell membranes, and it is the raw material for the synthesis of vitamin D, steroid hormones, and bile salts, and for bile salts. There's some fat-soluble vitamins we're going to go through, we're going to talk about as we progress in the semester. We'll look at A, D, E, and K. So some other fat-soluble vitamins. And we also have lipoproteins. They're responsible for transporting fats in our blood. Very important for us. So here we can see cholesterol. And here we can see testosterone. And then here we have estrogen.